Hello, and welcome to today's webinar. On behalf of Drug Target Review and DiscoverX, I'd like to thank you all for attending. I'm your moderator, Steve Bremer, editor of Drug Target Review. Today's speakers will be Laura Munro, scientist, R&D and Bioanalytical Technical Services at Sartorius Stedim BioOutsource, Lisa Blackwood, scientist, R&D and Bioanalytical Technical Services at Sartorius Stedim BioOutsource, and Alpana Prasad, who's product manager at Eurofins DiscoverX. Following their presentations, we will move on to a live question and answer session where you can pose questions to today's speakers. Please remember, you can submit questions at any point during the webinar using the Ask a Question panel situated below the player. So without hesitation, I will pass over to today's keynote speakers, Laura Munro, Lisa Blackwood, and Alpana Prasad. Not to worry, I wonder if we should perhaps start from the beginning because the line was very bad right from the outset. If that's okay.
Oh, gosh. Okay. Sure.
Okay, great.
Okay, so I just would like to thank everyone again for taking their time today to join us for the webinar on implementing ADCC assays in QC, where the donor variability isn't an option. The webinar will focus on the ongoing qualification studies at Sartorius using the single donor-derived primary CD16 effector cells. Before I hand over the webinar to Dr. Laura and Lisa, for them to share their study data with you, I would like to give you a quick overview of the KILR CD16 effector cells. So as I was mentioning earlier, that the therapeutic antibodies achieve their clinical efficacy not only by binding to their target antigen, but also from using the Fc domain-mediated recruitment of the immune effector cells in order to kill the target cells. Therefore, the developers of the therapeutic biologics must assess all possible mechanisms of action for their molecules, including antibody-dependent cell-mediated cytotoxicity, or ADCC, which is one of the most important mechanisms for targeting tumor cells. There are several approaches to measure ADCC activity, including the traditional methods in which the target cell death is measured directly, or with surrogate methods that use engineered reporter cell lines to measure ADCC potential. The regulatory agencies require that the antibodies that use ADCC as one of the mechanism of action, the cytotoxicity assays used in their release methods, are reflective of their MOA and use biologically relevant measures of the target cell death. Now, as you know, the classical or the traditional ADCC assays that are mainly the radioactivity or dye-based assays, they measure the killing of target cells by using isolated primary Pbmcs or natural killer cells, or the engineered NK cells as the effector cells. The success of these assays is highly dependent on the quality of these effector cells, and there we go. And hence present several drawbacks in the implementation for QC lot release, such as availability of fresh blood to isolate adequate amount of cells for running the assays when needed, as well as in the long-term support to be able to support the drug through its lifetime. Most importantly, the isolated primary Pbmcs or NK cells suffer from inter-individual or donor variability with low signal to background ratios. The challenge of variability can be addressed, and it has been addressed in some of the labs with the CD16 overexpressing engineered NK cell lines. However, these cell lines are known to be challenging to grow and often show high background lysis in susceptible cell models, as well as functional variability under different culture conditions. Additionally, a license fee is also required for use of these cell lines with marketed drugs, adding a significant cost burden to your development programs. So for implementation of these assays in QC lot release, a reliable source of effector cells is a critical consideration in order to achieve a consistent and robust assay performance with minimal lot-to-lot variability. So, to address this need for consistency and to reduce the assay variability, DiscoverX have introduced the KILR CD16 effector cells, which are primary effector cells that have been derived from a single donor. These are cytotoxic T lymphocytes that have been engineered to stably express the high affinity variant of CD16. The bottom panels show the phenotypic characteristics of these effector cells using flow cytometry. So if you look at the bottom panels, these effector cells are polyclonal population of 98% CD8 positive T cells, and these cells retain their T cell phenotype, and as this middle panel shows, over 98% of these CD8 positive cells are CD3 positive. The last panel shows the homogeneous and the stable expression of CD16 on these effector cells. Also, which is an important consideration for a QC implementation, is that these effectors are manufactured in quality environment for us to be able to ensure lot-to-lot and intra-lot consistency. Okay, to the next slide. Okay, so as I mentioned, that these are engineered T cells that mediate ADCC via CD16. So if you look at the ADCC mechanism, the left panel shows the classical ADCC model where the NK cells, which express the Fc gamma receptor known as CD16, engages the Fc portion of the antibody. The antigen-specific Fab region of that antibody engages the tumor cell. This antibody-mediated engagement of the tumor cell derives the NK cells to release perforin and granzyme that results in lysis of the tumor cells. The KILR CD16 effector cells mimic the NK cells where the expressed CD16 mediates the interaction with the antigen-specific antibody on the target cell, binding to target cell, and then triggering the cytokine release that results in the target cell death. So what this approach does, it removes the non-specific background killing that is characteristic of the NK cells and provide a very simple and a clean platform to measure ADCC as a function of the potency of the antibody. So the next question is, why these engineered T cells, why use these engineered T cells in place of the engineered NK cells, especially when you're looking to overcome the variability? Now, NK92 cells, which are often used as an alternative to the isolated primary cells to overcome the challenge of variability, these cell lines do offer significant challenges for their implementation in a QC environment. As I mentioned earlier, that these cell lines are difficult to grow and they deliver high non-specific lysis of the target cells. So the data panel on the right shows a relative background lysis with the NK92, which is the blue bar, as compared to the KILR CD16 cells, which is shown as a pink bar, with two different target cell types. As you can see, the NK cells have a much higher background killing, even with the killing-resistant SCOF3 cells compared to the KILR effector cells. Similarly, this is something that we have seen across several other killing-resistant cell lines where the NK cells or NK92 cells show a significantly higher background lysis. So these KILR effector CD16 cells are universal effector cells that can be used in any ADCC application that measures target cell depth directly. Whether you're using the KILR target cell lines or your own in-house generated target cells, you just need to simply replace the immune effector cells, whether they are the Pbmcs or NK cells, with the CD16 effector cells. However, we recommend that you reduce the ET ratio by 2 to 4-fold as the starting point, since these effector cells are fairly potent killers. So when you're using these effector cells in any cytotoxicity assay, these are first thawed and put into the media to recover for at least 24 hours before using them in an assay. The cells are then added to the target cells that have been opsonized with the antibody. The antibody-mediated cell death can then be measured by the appropriate detection method specific to the assay. This particular example in the slide just shows the use of killer target cells where the cell death is determined by measuring the amount of killer housekeeping proteins released due to the target cell death. As you can see, it's a fairly easy swap of the effector cells with the new KILR CD16 effector cells for any of your ADCC assays. Okay, so as we are focusing today on implementation in a QC environment, an important consideration is the ability of the assay to discriminate small differences in the antibody activity due to minor variations during production. When compared to the most routinely used effector cells, the Pbmcs, the KILR CD16 effector cells deliver significantly larger assay window with higher signal-to-background ratios, which provides a much better platform to evaluate ADCC activity of the antibodies. The data presented here shows the assay windows with 3 different types of the effector cells in the rituximab model, which is an anti-CD20 antibody, and also with a daratumumab model, which is an anti-38 model using the Raji cell pools. As you can see in both of these models, even when you're using the same ET ratio of 10:1, the KILR CD16 cells deliver a significantly higher signal to background and Emax relative to the NK cells, and also a 6-fold higher S/B or signal to background when compared to the Pbmcs. So, and another consideration in the quality environment is the lot-to-lot reproducibility for the assay components. This slide shows 4 different production batches of the effector cells, which have been manufactured over a span of 2 years and were tested for ADCC activity in a single experiment using the anti-human CD20 model. The data demonstrates a very low lot-to-lot variability and no significant variation in the killing capacity of the cells in killing capacity of the CD16 effector cells compared to the Pbmc isolated from different donors, as data presented in the 2 tables to the right. This excellent reproducibility demonstrates the ability of these cells to deliver consistent and reproducible performance throughout the life of the drug from development to post-market. So another critical consideration when selecting and implementing an assay in QC is the reproducibility between the analysts. This study, the data shown here on this slide, included a study for using 2 analysts who taught 2 different vials of the effector cells and performed the ADCC side by side. The data demonstrated here shows very low inter-user variability, making these cells highly amenable to implementation in labs across several sites of your organization or to your CRO partners. So to summarize, some of the key benefits of using KILR CD16 effector cells for your cytotoxicity assays. As I mentioned, these are universal effector cells that are compatible with any ADCC activity that directly measures target cell death. These primary effector cells are derived from a single donor, thereby eliminating the donor variability that reduces the reproducibility of the ADCC assays. These are cryopreserved, ready-to-use cells, which allow the customers to easily implement these in any lab from discovery all the way through assay development and to QC. The higher signal-to-background ratios compared to Pbmcs and an excellent intermediate precision as well as lot-to-lot reproducibility makes them ideal for use in long-term QC testing. These cells measure the target cell death, which is the biologically relevant measure of ADCC. So with this overview of the KILR CD16 effector cells, I would now like to hand over to Dr. Laura and Dr. Lisa at Sartorius Stedham, who will now share some data from their ongoing qualification studies using these effector cells. Lisa, over to you.
Thanks, Alpana. So today we will be discussing the following: an assay format overview, the CD20 expression on Raji cells, Our approach to qualification design, as well as the results for qualification studies using the KILR CD16 Effector Cells and an enriched NK effector cell, followed by a comparison of the two studies. In addition to this, we will look at a case study into the use of KILR CD16 Effector Cells and an NK enriched effector cells using an infliximab innovator and biosimilar. At Sartorius Pbm Bio-Outsource, we have a breadth of knowledge and experience in the development of off-the-shelf assays for characterization of monoclonal antibodies. All IgG1 antibodies have the potential to cause ADCC, and therefore assays to measure ADCC activity are an integral part of our testing services for biological products. DiscoverX target cells contain a tagged reporter protein which is released into the media when the cells have been compromised due to cell death. Our effector cells are typically an enriched NK cell population isolated from fresh human blood. For this work, we substituted our primary Pbmcs with the KILR CD16 effector cells to assess ADCC activity. This was the only change we made to our predefined protocol. Here, we thaw the KILR CD16 effector cells and allow them to rest for a period of 24 hours, and then up to 7 days. The target cells, in this case RAGI cells, are seeded at a known density, then a dilution series of monoclonal antibody is applied before addition of the effector cells, and the components allowed to incubate for an optimized period of time. The endpoint of our assays uses a luminescence detection reagent to measure cell death as shown in this diagram. 20 years ago, in November 1997, rituximab became the first monoclonal antibody to be approved for the treatment of cancer. Rituxan in the US, or MabThera in the EU, is now an approved treatment for non-Hodgkin's lymphoma and rheumatoid arthritis, amongst others. Monoclonal antibodies mediate anti-tumour effects by a variety of mechanisms resulting in cell cycle arrest, direct induction of apoptosis, complement-dependent cytotoxicity, and of course ADCC. The primary mechanism of action for rituximab is ADCC by targeting CD20, a protein found primarily on the surface of B cells. We assess the expression of CD20 on the surface of RAGI cells using the IQ Screener Plus platform from Intellicyte. This high-throughput flow cytometer is an integrated instrument, software, and reagent system that enables rapid, high-content, multiplex analysis of cells and beads in suspension. On the right, we have a histogram plot showing binding of an RPE-conjugated IgG secondary antibody bound to rituximab on the surface of RAGI cells. Shown in blue is the unstained cells. The shift of 2 logs to the right shows expression of the CD20 receptor. This, in combination with a narrow peak, indicates a consistently high level of expression within the population of cells. We have therefore shown that the RAGI cells are a good target for use in assessing rituximab ADCC activity. Now to come on to our qualification study design. ICHQ2 states accuracy, intermediate precision, linearity, range, and specificity should be evaluated when validating an analytical method. At Bio-Outsource, we use these guidelines as a basis for qualifying our off-the-shelf assays for biosimilar compatibility studies. Accuracy is assessed at known concentrations relative to the reference standard over the range of 50 to 200%. This ensures that a wide range can be tested in our assays and allows us to support clients from clone selection through definition of critical quality attributes from an innovator population right through to finished product compatibility studies. We set up the following qualification study to allow us to assess the major requirements of ICHQ2. Over the course of the study, we used 2 different operators, 3 different vials of KILR CD16 Effector Cells, and the assays were performed over 3 separate occasions. This allowed us to assess the following: intermediate precision at 50, 100, and 200% on different days using multiple operators and effector cell vials. Specificity controls are of the same isotype as the molecule undergoing assessment but are not expected to elicit a positive response. The same vial of killer cells was used to test two samples at a nominal concentration of 100%, with two different operators performing the assays. The third sample at 100% was tested using a second vial of the KILR CD16 Effector Cells with a single operator. This showed that interoperator precision was very good, with the relative potency result being within 10% of the expected value when different operators used the same reagents. The magnitude of the response was 3.4 and 4.1, with the curve shapes being highly similar. The slope values were 0.8 and 0.9 respectively. This also showed that precision between vials is consistent as well as accurate, with the mean concentration of 100.0% returning a percent CV of 11. The accurate and precise results obtained indicate that the KILR CD16 cells would be suitable for use in assays that required a high degree of reagent control. These would include assays such as lot release or stability studies due to the low variability and the relative potency results generated between different operators on the same day. In addition, the results generated using different vials of the cells thawed on different days also shows good correlation between the potency results, demonstrating robust assay performance. As well as the accurate relative potency results returned for the 100% samples, the relative potency results generated for samples tested at the nominal concentration of 80% and 125% relative to the reference standard also showed accurate results. The potency values, as shown in the table returned, were within 2% of the expected value, further demonstration of the suitability of the KILR cells for both lot release assays and for compatibility studies for biosimilars. In the following table, you can see the results from our qualification study. Across the range of concentrations tested, 50 to 200%, there is no pattern in the level of inaccuracy observed for the multiple samples assessed, indicating that there is no inherent bias in the assay. This, coupled with a maximum inaccuracy of 20.8%, suggests that the KILR cells are suitable for assessing potency across this range in an ADCC assay. The most inaccurate results were at the extremes of the concentrations assessed, minus 20.8% at the 50% sample and minus 15.8% at the 200% sample concentration. At samples closer to 100%, between 70 and 143%, the maximum inaccuracy observed was just over 12%. The data generated for the accuracy assessment was used to assess the linearity of the assay, resulting in an R-squared value of 0.9989, which indicates that an accurate result would be obtained over the range of the assay. In conclusion, the KILR cells demonstrate good alternative to isolated Pbmc preparations from blood and could be used from clone selection and process development to compatibility studies as well as lot release assays. Effector cells for use in ADCC assays are traditionally isolated from whole blood, which requires availability of donors with the required genotype. These assays are often highly variable due to donor-to-donor variation. This can be reduced by using selective pools of genotype donors. However, there is still an inherent variation which we don't want in our assays. Isolation of Pbmcs from blood is a complex process. Firstly, blood is drawn from a donor. This is then separated using lymphoprep into several layers. The Pbmc layer is then isolated and enriched NK cells collected. Then a dilution series of the monoclonal antibody is applied before addition of the enriched NK effector cells, and the components are allowed to incubate for an optimized period of time. Again, the endpoint of these assays uses a luminescent detection reagent to measure cell death. In a study performed here at BioOutsource, we investigated the expression of CD16A from the isolated NK effector cells across 9 different donors and with the 3 different genotypes. These cells were isolated in a single day, reducing variation from other external sources. However, you can see from the graph there are still differences in the percentage of cells positive for CD16A expression, ranging from around 60 to 90% enrichment. This is a consequence of variability in the ADCC assay, Therefore, enriched NK cells would not be suitable for use in QC lot release assays. The process is simplified when using the KILR CD16 effector cells and reduces the inherent variability associated with traditional methods. Here we show representative data for the nominal concentration of 100% in the enriched NK cell ADCC assay for rituximab. This was tested using two different genotypes, VF and FF, two different operators performing the assay over 3 different days and in 2 different laboratories. Although the mean relative potency results are within 10% of the nominal value, there are some differences in the EC50 values between FF and VF donors, which is expected from the differences between the genotypes. There is good inter-operator precision and consistency between results over different days in laboratories. However, there is a higher failure rate due to low responding donors. Overall, this assay was deemed to be accurate and precise across a wide range of concentration and is ideally placed to be used within clone characterization and similarity studies. When comparing both methods, there is not a large difference in precision, accuracy, or dilutional linearity between the KILR CD16 effector cells and enriched NK effector cells. Both methods provide good accuracy and precision across a wide range of concentrations and correlate well with each other. The fold response is greater with KILR CD16 effector cells compared to purified NK effector cells in the rituximab ADCC assay, which may provide a more consistent assay window for assessing parallelism. When used in the rituximab ADCC assay platform at BioOutsource, the KILR CD16 effector cells perform comparable to the qualified enriched NK effector cell for use in characterization and similarity studies. However, using the KILR CD16 effector cells would represent a more suitable option for QC and lot release assays. I will now pass over to my colleague Laura to talk about our case study using infliximab.
Thanks, Lisa. So, having qualified the KILR CD16 effector cells in rituximab ADCC, we used them in a case study looking at sensitivity to differences between innovator and biosimilar molecules. In this case, infliximab, an approved treatment for Crohn's disease, rheumatoid arthritis, and psoriasis, among others. The antibody works by blocking the action of TNF-alpha by reducing inflammation. The binding and function of an antibody is influenced by the structure and glycosylation of the molecule. The physical-chemical analysis are the initial tests that are usually performed. At BioOutsource, we have the capabilities of looking at the structure of monoclonal antibodies and determining their intact mass and glycan profile. We use these methods to investigate the difference in the glycan profile of these that have been expressed in mammalian cell lines. The profile of infliximab innovators and biosimilar molecules, and investigate the sensitivity of the CD16 effector cells to any differences. One approach to the released N-glycan analysis is using PNGase F to release the glycans from the antibody. This is followed by the derivatization of released glycans to facilitate their analysis by liquid chromatography, mass spectrometry, or other techniques. Our approach is to perform the release N-glycan assay using rapid flow or MS label. In addition to a strong fluorescent response, its enhanced ionization efficiency compared to traditional labels such as 2AB leads to improved MS sensitivity. Following a solid phase extraction cleanup step, the released glycans are analysed by LCMS. This involves a high-resolution chromatographic separation of the glycans with online fluorescence and accurate mass detection. Each peak corresponds to a different labelled glycan. Most peaks in the biosimilar chromatogram shown in red are observed in the innovator chromatogram shown in blue, although there are some variations between the profiles. There are higher levels of sialic acid species and increased galactosylation of G1F and G2F. Both profiles show similar levels of high mannose, however there's decreased afucosylation in the biosimilar profile. This decreased afucosylation can have an impact on ADCC activity. The ADCC process is mediated by the Fc gamma receptor 3A, or CD16A. This is expressed by natural killer cells and functions to recognize and bind the Fc portion of IgG antibodies. Two distinct allelic variations exist for this receptor, giving rise to three different genotypes: VV, FF, and VS. Each with different affinities for IgG, and clinical differences have been reported between patients of different genotypes. The results obtained from ADCC assays can also be affected by different Pbmcs obtained from donors of differing genotypes. As part of early phase ADCC evaluation, therapeutic monoclonal antibodies should be evaluated against a battery of donors with different genotypes. As mentioned, the structure of an antibody affects its binding and function. Using an MTNF expressing cell line, we investigate the sensitivity of the killer cells. Sensitivity of the killer cells compared with VV and FF genotypes purified from NK cells to the differences in glycan profile of the infliximab innovator and biosimilar molecules. The following potencies were returned during the assay. The VV on the graph of the top left returned a relative potency of 29.7%. The FF at the top right returned potency of 38.7%, and the killer cells 41.0%. This indicates that the killer's CD16 effector cells behave similar to the FF donors in the infliximab ADCC, and we are able to detect the differences in glycan profile between the molecules. So the benefits of using the KILR CD16 effector cells are reduced variability and reproducibility between analysts and on different days. They can be used for QC lot release due to low variability with good accuracy and precision. They are not dependent on genotype. Use of the killer cells reduces the reliance on supply of fresh blood, and it is a rapid assay format when compared with traditional ADCC methods. It also allows us to support clients from clone selection through to lot release of their product, and they can be used with a number of different target cell lines which are specific for your antibody. And they are sensitive to differences in the glycan profile. Thanks for listening, and we will be happy to take your questions. If you'd like further information about KILR CD16 Effector Cells, please contact DiscoverX. Thank you for your time.
Thank you, Laura, Lisa and Alpana, for your excellent presentations. Now, before we move on to our live question and answer session, I would like to invite you to provide us with some feedback on today's webinar. In a moment, a poll will appear on your screen asking you to rate the webinar. If you could take a moment to provide your feedback, that would be much appreciated. We now have the pleasure to introduce our question and answer session, where today's speakers will answer all your questions. Don't forget, you can submit your questions using the Ask a Question section situated below the player. Now, let's take a look at the first question. Okay, this one's for Laura or Lisa. Would you please address the issue of hypersensitivity of some engineered ADCC lines, particularly reporter gene constructs, to small afucosyl and high mannose glycan differences between AB drug lots and the sensitivity of this system to small glycoform differences?
Okay, so I think this one was covered in the infliximab case study. In terms of the killer cells, we've shown that they're sensitive to differences in the glycan profile. If I just go back to one forward. This one here, sorry. So we've shown that they're sensitive to differences in the glycan profile of the infliximab innovator and biosimilar molecules. and the data in this slide suggests that they show the same level of sensitivity to the FF donor, but they can detect the differences. So I don't think that there's a hypersensitivity that they're going to miss that they'll not be sensitive to differences in the glycan profiles here.
Okay, great, thank you. Here's another one for Laura or Lisa. How do we decide reference sample if there is not any international standard?
So firstly, you would need to define your critical quality attributes early on in your biosimilar program. You need to ensure that your innovator characterization is carried out during multiple steps throughout the whole development process. One of the main things is it's really important to source lots from different geographies, and also to make sure that they have long enough expiry dates with sufficient quantities to ensure that this can carry you through your whole biosimilar program. You would never choose just one reference product. You need to choose something that's representative of whatever is available on the market. So you would choose a whole bank of innovator products to compare your biosimilar to, so that you ensure that it's representative within the range that you're going to find in the market anyway. So you should never ever just choose one. You should be able to choose a whole bank. We would suggest between 10 and 30, depending on the availability of your product.
Okay, thank you. Next question, again to Lisa or Laura. How do you compare engineered NK usage in ADCC assay compared with NK cells isolated from Pbmc? And which one is more suitable for QC standard?
So I think we showed again in our infliximab case study that the KILR CD16 cells are compatible with an enriched NK cells from Pbmc preparation. In this case, the KILR cells behave similar to an FF donor, indicating that perhaps they aren't as sensitive as a VV donor. However, these donors are high responders. So the KILR cells are still representative of what you would find in the population as a whole, and it's much better for use in QC lot release because there's much less variability since you only have the one donor. So there shouldn't be any issues with using the KILR cells in comparison to NK cells isolated from Pbmcs, and definitely better for use in QC lot release studies.
Okay, here's another question just come in. Are these cells already accepted by the authority as a replacement for NK/Pbmcs?
I'll take that question. So just to let you know that DiscoverX introduced these KILR CD16 Effector Cells just 3 months ago. So they are fairly new to the market. We expect, since these are getting used in the development and implementation now with some of the QC assay development programs, we should start seeing some feedback from the regulatory authorities for the use of these cells. And we will keep you all posted. Thank you.
Thanks, Alpana. Next question. Can you guarantee that the cells from the single donor are available for the next few years and in an adequate amount? Will it be necessary to change to a new single donor necessary in the future?
Okay, I'll take that one as well. So the way we have built the supply chain for the cells from single donor is that we have extensive and a large number of master cell banks that are available from which we generate the working cell banks. So the availability, as we will support the development programs is based on your requirements of the number of cells, the assays that you're running, and how frequently. We would be reserving the banks for you exclusively so that you're able to feel assured, and then we are able to ensure the supply for the lifespan of the drug, which could be anywhere from 10 to 15 or even longer years for your drug. And as far as in changing to a single donor, the technology that's been used to generate these assays can be used to generate a bank from another donor. However, we have a sufficient supply for now that we do not need to generate another bank from another donor.
Okay, thank you. Next one. How do regulators look towards reporter ADCC assays?
So based on the presentations or discussions that we have seen at the different conferences on bioassays and the ADCC, the discussion had been that the sponsors who are looking to submit a or include a reporter assay in their dossier to the regulatory agencies have also had to do a bridging study to be able to demonstrate that their antibodies is indeed able to kill the target cells, because killing is the endpoint of a mechanistically as well as biologically relevant readout. So essentially, if you are looking to submit or include a reporter assay, you may need to also generate a bridging or a supporting study to show that the antibody does indeed kill and is stability indicating enough to be able to discern the differences during the production of your antibodies post-marketing.
Okay, thank you. Next question. Is an ADCC assay required for batch release?
This would basically be determined if the ADCC is the main MOA of the molecule. So if the ADCC assays are going to be implemented as a lot release assay, the ADCC has to be the MOA of the biologic and also be a stability indicating, as I just mentioned earlier, that the assay should be able to discern the differences in the killing capacity or killing potential of the the biologic or the antibody through the production post-marketing.
Okay, thank you. Another question here. Are these Pbmcs? That is, do they include CD3 plus T cells or only CD16 effector cells?
So if I could go back to, I think it was my third slide. Just give me a moment. So essentially, the CD16 effector cells are cytotoxic T lymphocytes. These have been enriched from the Pbmcs from the donor, and these are these CTLs, which are CD8 positive, retain their T cell phenotype, which is CD3 positive. I still am far away from my slide. Okay. Here we go. So as the bottom panels, which are essentially showing the phenotype of the CD16 effector cells, you can see these are cytotoxic T lymphocytes with the greater than 98% CD8 positives. And these retain their TCL phenotype, which is when you're looking at the CD3, which is 98% CD3 positive on the cells. So these are not Pbmcs alone, because Pbmc is a heterogeneous population of multiple different types of cells. So these have been enriched and are polyclonal population of the cytotoxic T lymphocytes. And also to add, I'm sorry, Steve, I just remember the other part of the question is that are they only CD16 effectors? So they have not been isolated to enrich for CD16 effector cells. They have been enriched for CD8 positive cytotoxic T lymphocytes, which have then been engineered to express CD16s.
Okay, next question. How would you automate this assay in a GMP compliant manner?
We haven't yet received, or we haven't had any discussions at this time about automating the assays. However, the assay platform, if you're currently using your classical assays in an automated setup, you should be able to simply swap out your effector cells with the CD16 effector cells. As far as the KILR platform goes, that's a very simple and a linear platform as well, and it's a homogeneous assay that you can also automate as well.
Okay, thank you very much. Unfortunately, that's all we've got time for today. Any unanswered questions will be responded to via email. So please join me in thanking Laura Munro, Lisa Blackwood, and Alpana Prasad for their time today. And on behalf of Drug Target Review and DiscoverX, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.